Good morning, good afternoon. My name is Steve Van Warmer. I'm the president and CEO of PAH Global Association. We're very excited to have you here with us today for our ongoing series on air with PH Aware seminars, webinars about exploring the remarkable evolution of pulmonary arterial hypertension from its earliest challenges to today's breakthroughs and the promising future of research and care. Whether you're a patient, a caregiver, healthcare professional, researcher, or advocate, these live conversations offer valuable insight into how far the PH community has come and where it is headed to next. We're really thrilled to have Dr. Stephen Chan with us here today as he examines the next generation of PAH therapies and the scientific breakthroughs redefining what's possible. And as we learn about basic, translational, and clinical research and how they are converging to identify novel therapeutic targets, accelerating drug development, and shaping the future of PH care. Dr. Chan is a physician scientist and professor of medicine at the University of Pittsburgh School of Medicine, where he directs the Vascular Medicine Institute and Center for Pulmonary Vascular Biology and Medicine. His research focuses on uncovering the molecular mechanisms driving pulmonary arterial hypertension, translating those discoveries into new therapies. Dr. Chan is an internationally recognized doctor for his work on emerging PAH targets, including metabolism, inflammation, non-coding RNA, mechanosensing, and cellular biology, with the goal of developing treatments that go far beyond today's established therapeutic pathways. Following his presentation today, we're going to have a lot of time for your questions, so please feel free to put those in the chat feature at the bottom of your screen or in the Q&A tab as well, and Dr. Chan will answer those after his presentation. And I want to say I first met him. I saw him at a standing room only presentation at ATS a few months ago. So we're really thrilled to have you here today as we kind of continue this exploration into where PAH treatments are going. Finally, I want to say a big thank you to our webinar sponsors today, Merck and Liquidia. Through the strategic use of innovative technology, and compelling storytelling, your support for PH Aware helps us amplify the voices and experiences of patients worldwide, fostering a more informed and connected worldwide community. So thank you again. And without further ado, welcome to the future of PAH, emerging targets and therapies. Dr. Chan, again, thanks for being with us today. I will let you take it from here.
Well, thank you very much, Steve. Really appreciate the opportunity. Very thrilled and very privileged to be here. Hopefully, we can have a good discussion. As Steve said, I have a few prepared slides that I'm going to give, but a lot of time afterwards for questions. So I really do invite you all to ask questions. Would love to have a conversation with you all about the future, which I think is going to be very exciting. Ms. Schweitzer says there is a lot of echo. Do you all hear an echo? Steve, do you hear an echo?
I don't hear an echo. I don't know if, let me see if Michelle, I think Michelle, it says that you are logged in twice. You might have two logins kind of simultaneously going. You might want to look at how you're connected. I see you on here twice.
Great. Okay. So hopefully we can have that corrected. But I'm going to continue. We're going to try to share the screen and start going on the formal part of the presentation. So let's see.
You're good to go.
Do you all see the screen, Steve?
Yes, sir. You're good.
All right, very good. So, today I have the opportunity to talk about my view on the future of pulmonary arterial hypertension, talking about emerging targets and therapies, talking about new research and new research directions. Now, this is a pretty exciting time, but a very daunting task in this regard, because everything I say here is going to be based on my personal prediction and could, of course, be totally wrong. But I've had a really great time thinking about what the next phase of research and patient management and patient care is going to be. And I think it's going to be a very exciting time, no doubt. And let's see whether or not some of these things can actually come to fruition. And so this is just my disclosure slide that I have some grant funding and consultations, and that I have founded a couple of startup companies called Synhale Therapeutics and Elysian Therapeutics as well in the space of pulmonary hypertension. Okay, so I'm going to, divide this talk into four major areas and structure this perspective around those pillars. So from my biased view, I think that science, medicine, clinical care are going to be consolidating around four major areas where I think that the opportunities for discovery and applications to clinical management are going to be important and evolving. So the first concept that I'm going to explore is the idea that basic science, molecular science, translational science, and clinical research and clinical care in PAH are converging in ways that we've never seen before, and it is extraordinarily exciting. Typically, we often think about the science as very siloed and isolated away from what we do in terms of clinical trials and away from what we actually do in clinical practice. And I think that now is an opportunity where all of those stars are aligning, and we're finding that there's a lot of overlap, synergy, and complementarity in that perspective. I also think that we are going to be seeing a lot more and understanding a lot more about pulmonary arterial hypertension beyond just the heart and lungs. Historically, we've thought about this disease as sort of a heart and lung issue. But now we are actually realizing, and perhaps our patients have been pushing us to think about this more, that there are other manifestations of this disease that go beyond the heart and the lungs. It goes into exercise and skeletal muscle. It goes into the brain. It goes into the kidney. It goes into the liver. And we're seeing a lot more than that in that regard. And I think that we're also going to be understanding a lot more and have insights into how our organs communicate with each other, and that's going to shape discoveries in PAH. In terms of therapies, we are already seeing a new advent of next generation PAH therapies, and that's going to push us further into understanding this disease more and understanding how we can better serve and treat each individual patient as compared to a one size fits all, every patient gets the same exact medication. So, we're going to talk about the idea of precision medicine or individualized care at a much more molecular level in the future. And finally, I want to touch upon the idea that technology aided processes, that is to say, through artificial intelligence, through bioengineered opportunities, these are all accelerating innovation in ways that we have not seen before, and I think are going to be very, very important in the future. All right, so for the first pillar, I wanted to talk about this idea of converging scientific disciplines. So the converging of basic science with clinical science in PAH. And I'm going to use aging research as an example. Now, over the past 20 years, we have been witnessing a shift in what we are seeing in terms of the prevalence of pulmonary hypertension and pulmonary arterial hypertension to a much older population. So this is reflected in these two graphs shown here on the left, one from the Compara registry and one from the Aspire registry. These are both European registries that have been following patients over the past 30 years. And as the graphs show, instead of actually seeing just the 18 to 40-year-olds that we had historically seen for idiopathic pulmonary arterial hypertension when it was first being treated and described in the 1960s and 1970s, we are now seeing a much higher percentage of the population in the fifth, sixth, and seventh decades of life. But the problem is that we do have very little knowledge of the molecular and even the clinical insights that are surrounding people who are older, who have pulmonary hypertension and have manifestations at these stages of life. I'm going to touch briefly on the idea that in basic science or molecular science, there is a renaissance right now in terms of what we think is happening in the aging population. As we age, what's happening with our cells and tissues and molecules. And this particular picture is actually a very famous one that a lot of scientists know very well about the hallmarks of aging. There are certain processes that occur at the cellular level that happen. We've been thinking about that a lot in neurobiology, in cancer, and in aging in general, but we haven't really done so as much in pulmonary vascular disease, pulmonary hypertension. Some of the key items are shown here in terms of these words surrounding it. Ideas like inflammaging. So the combination of both aging and chronic inflammation is driving disease. The term senescence, that at a cellular level, our cells are now changing at an older age where they're not replicating themselves. They're not proliferating at such a fast pace. That happens as we get older. That is also influencing how pulmonary hypertension develops. Metabolism, how we utilize and store energy, that's also a very big deal in aging and how that actually operates in pulmonary hypertension. A couple of other things here on the right: genomics, epigenomics, lysosome biology, some jargon here, but essentially, how does our DNA and the way the DNA is structured and operates in the aging cell, how does that actually affect the way in which pulmonary hypertension evolves? Those are things that we're going to be seeing much more in terms of discovery processes, and I expect that these efforts to understand them are only going to intensify in the future. Now, I believe, however, that just the basic principles of cellular and molecular aging are only one part, one half of that story. Because now we are at a point where we can connect those basic principles to exactly how our patients, how you all, how I experience life over the decades of time that we are on this earth. And a great example of that type of study is the impact of the cumulative exposures that we see in life and how that relates to pulmonary hypertension. So I'm going to talk about this idea called the exposome. Now, the exposome is a term, another jargon term that we as scientists utilize that describes numerous and often chronic exposures that we experience that could be significant to both our health and disease, particularly as we age. And these can include our own personal ecosystems, our lifestyles, our social interactions, our physical and chemical surroundings that we experience. Work in pulmonary hypertension is accelerating across all of these different types of exposures in many different ways. And that is in terms of the diet that we eat, the pollution that you are exposed to, the anxiety and the stress that you are exposed to just in the decades of life that that we are alive, the climate and the climate changes, contaminants in our food, in our medicines, and the creams that we put on our body, sleep and circadian rhythm, and exercise. All of those things we know are important and we know that they affect us, but we really do not understand as much how they relate to the molecular and cellular changes of aging and how they relate to the pulmonary vasculature. So I envision a future where basic science in pulmonary hypertension now can be directly incorporated into what we call epidemiologic analyses of our patients with pulmonary hypertension over time. This is a picture of a person who's wearing a couple of wearable technologies. The watch that many of us utilize could be a ring, could be something that's around our head that really, in real time, can track many different aspects of our behavior across diet, sleep, cognitive function, exposures, molecular profiles, and so forth. And with all of that data coming out across days, weeks, years of time, we can then utilize machine learning and artificial intelligence in order to harmonize the data and to understand essentially what are the stressors that we are experiencing, how they relate to our own genetic makeup, how they actually lead to adaptation, recovery, and hopefully something that we would really like to focus on more in the future: resilience to pulmonary hypertension, and how can we best exploit that and make sure that that is optimized in all of our patients. So I think that that's something that we're going to be seeing more in terms of clinical observations. You may actually be even asked by a number of your PH specialists to wear a watch or to wear a ring that will track these sorts of things for us to be able to get better understanding under those circumstances. And I think that that future is coming at us fast and furious. And I think that that's going to be a very special time for us to understand this disease at a much more holistic level. All right, so that's the first pillar. Second pillar I'd like to concentrate on is this idea of inter-organ communication. that we are seeing that pulmonary hypertension is much more than just heart and lung disease, that we may be seeing manifestations at a syndromic level. Syndromic meaning that many different tissues and organ systems are affected than just the ones that are in our chest. And that could be across the liver, intestines, fat, kidney, skeletal muscle. And our group is especially interested in brain and cognitive dysfunction that we often see in our patients with pulmonary hypertension. Now, I don't have time here to go into the nitty-gritty of some of that science. If there are questions about that, I have a lot of great detail and information about some of the work that we're doing in terms of the brain heart lung connection that we're seeing in pulmonary hypertension. But mainly what I want to address is that if this is the future, that is to say that if we really want to understand how the body holistically is experiencing and allowing us to understand sort of what is going on with pulmonary hypertension, we need to do a better job in terms of how we collaborate across different types of science and across different types of medicine. So an example comes from our pulmonary hypertension center here in Pittsburgh, where I direct our center that not only is involved in heart and lung issues of pulmonary hypertension, but we have established a growing collaborative partnership with a lot of different centers, programs, divisions, and departments that go far beyond the heart and lung and will actually be very important in understanding how does this disease affect everything within our body across time, space, and your lifetimes. So I think that that's really important to think about, and I think that's going to be a different model of what we've typically seen in how pulmonary hypertension research is pursued. All right. The next pillar that I want to address is this idea of next generation therapies. Many of you have already known that there are new medications that are being tested, new medications that have been approved by the FDA that are in many ways very transformative, that we're just learning about right now, that you are experiencing as well. And I think that that sets us up for a new future. It requires, though, a more precise understanding of who will and who will not respond to these therapies and who are actually prone to side effects that we need to watch for very carefully. Now, a prime example is the introduction of sotatercept about a year and a half ago. Some of you might actually be on sotatercept, which is Winrevair. It's an activin signaling inhibitor, which we think is very central to the primary origins at a molecular level of this disease. We now know that there are emerging questions. There are emerging questions as to whether or not it's disease modifying or reversing. There are questions about, is it effective across all different types of pulmonary hypertension, or are some people with specific types of pulmonary hypertension more or less responsive than others? Is it pleiotropic, and in what ways? Pleiotropic is another jargon word suggesting it has a lot of different features, it does a lot of different things. So is it important not only in the blood vessels, but maybe it's also important in the bloodstream itself? Is it important in the brain? Is it important in the heart? I think that a lot of those things are still being worked out right now. And why is it that some people have a great response to it and others have a modest or a mild response to it, and some don't have any response to it at all? What defines the variable response, and how can we do better to understand that? I think another aspect that we're seeing as well are the effects of inhaled prostacyclin. Many of you are also probably being treated with inhaled prostacyclin, but we now are understanding that it has many more actions than just simply relaxing blood vessels. And we think, and we've been seeing clinical trials that are showing that it affects lung tissue in and of itself. It affects other areas of the body. And I think that that's also something, an example that these medications still have a lot of surprises up their sleeves. And I think that the more we understand, the better we're going to be to truly treat at an individual level. And so this sets us up for this idea that as we think about pulmonary hypertension, we often utilize this clinical classification of 5 different major groups. And that has actually, every 5 years, the global leaders in pulmonary hypertension will debate and will recommend who and who should not be in each of these groups. And this has actually treated us well so far. But I think that one of the things about the new era is that as we deploy more and more new and next generation drugs, we're going to be relying a lot more on genetic and molecular markers that represent the driving features of pulmonary hypertension. And so, very similar to the idea that if we can actually understand and get more data from the profiles of our patients across space and time, this is going to be helpful for us to even revamp this entire clinical classification system to perhaps a new classification system where we are going to be able to understand at a molecular level which patients will respond to which medications. And perhaps that's going to be important to be using medications across these different groups, but in specific scenarios when we know it will be the most effective and with the least side effects. And I think that that's going to be a very different time, different way of treating and thinking about pulmonary hypertension. It's going to be at a molecular level, perhaps, and not a one size fits all scenario, which we've been used to in the past. All right. Then the final pillar that I want to emphasize is that we are maybe finally on the verge of addressing what I've called a black box, meaning that something that has been a challenge for us to really understand very deeply what is happening in terms of development of pulmonary hypertension deep within the lungs. This has been a major problem because we don't typically get tissue to be able to study the cells and from you suffering from pulmonary hypertension until we are able to either get transplant tissue or, unfortunately, upon when a person dies. But the problem with that is that we only then would be studying the end stage of disease. We have very little knowledge about what happens at the very beginning of disease, so-called inception, and the development of disease over time. That is the black box. We have very little ability to get information in that regard from patients who actually suffer from the disease during those circumstances. And the animal models that we utilize historically have not been perfect. They don't fully replicate the type of disease that humans have. So that has been very important to note that, because of that, there have been challenges for us to understand what's going on, how can we treat it, what are those targets. The lack of human tissue in the course of the disease has been a huge barrier to discovery. However, now I think that we have this possibility that this deficiency could be addressed through bioengineered models, such as 3D bioprinting of organs and organoids on a chip that may be replicating organ and organ system communication to a level that we've never been able to address or model. And it would allow for a more systemic ability to manipulate complex variables that are not even possible in the human condition or in the animal condition. So this could be a very, very powerful way to understand pulmonary vascular disease at a precise molecular level. And this could, of course, be accelerated by more advanced computational and AI-related modeling of cellular and organ aging, particularly as we understand more about the multiple different molecular landscapes that are changing in the body as disease progresses. And as we merge these landscapes together with clinical and physiologic data over real time, that will allow us much more insight and ability to actually understand which targets to go after and when. And that's something that I think that in the future, we're going to have a lot of progress in. The final thing I want to mention, and again, this is a really big topic in general, is the idea of AI or artificial intelligence guiding discovery in pulmonary vascular disease, something that a lot of us are very excited about, a lot of us are actually looking at very carefully. That future is still murky, and I think that this picture kind of emphasizes the idea that maybe we are going to accelerate in this process. One possibility, however, has been touted is that, well, maybe AI is going to take over the entire operation. I would argue, however, that I very much believe that human creativity and guidance will still be very paramount here. The future of the AI taking over and leaving humans out of it, I think, is going to not be the future that we want. I think it would be the wrong direction, because I do think that there is a role, a very important role in terms of oversight, both at a clinical level, at a research level, and even at the patient level, to make sure that there is a human component to this, ethically, from a creativity standpoint, and from a guidance standpoint. This is really important to emphasize, and I think that this will be an accelerant to our discovery, but it will not take it over entirely. So, in summary, I guess I'd like to end by saying that I very much believe that there's going to be a renaissance of discoveries in our field that will only accelerate. It will take the right type of investment and strategies. I think that it's going to involve an integration of converging disciplines, of what I'm going to call whole body science, personalized diagnostics and therapeutics, and artificial intelligence to aid and accelerate discovery. I do want to mention, though, that that idea of investment is extraordinarily important. Right now, we're at a really interesting tipping point where I think that we all feel that there are huge innovations that are being made. And if we make the right types of investments, both from an effort perspective, strategy perspective, and financially, this is going to pay off in ways that we've never seen before, and at a pace and an accelerated pace that will be very, very thrilling to see, great for our patients as well. I think that we are also, however, in danger. We are in danger of making the wrong type of investments, of pulling back on investments towards health. That if that is not prioritized, I think that these types of innovations will be stalled, and it will be to the detriment of our patients. So I very much believe that we need a partnership across the community, our patients, the government, foundations, philanthropists, scientists, physicians, everyone's on track with the same mindset to get to this future for the sake of our patients. So I do very much believe in that. I think that it's an exciting time to be in this field. I think that the type of conferences that we typically go to as scientists and physicians, you can feel the energy in the room these days, where I think that maybe 5 years ago or 10 years ago, there was, of course, energy, but not the same type of energy that we're seeing today. I think that there's a real acceleration, a real inflection point of the discoveries being made, the drugs and the medications being discovered and being tested at this point. And I think that there's going to be a huge influx into the field, and a really exciting time for our patients to be able to have those individualized, more effective therapies in the very near future. So with that, just end with this slide that, of course, a lot of this is my lab, but a lot of features about this type of innovation really takes a village. It takes a lot of different types of experience and opportunity to be had. We are very interested in making sure that this continues, particularly in terms of training our junior investigators and clinicians to be the next wave of innovators in this field, to ensure that our patients really are benefiting from these discoveries in the very near future. So with that said, I'm going to stop the sharing, and happy to answer any questions that you all may have now and, of course, in the future.
Awesome. Oh, thank you so much, Dr. Chen. That was great. One thing that just leaps to mind in that one of those last slides that you showed about just where we're at, or where we're headed. Do you think that with all this innovation and new things that are happening and new ways of thinking about this, new ways of modeling that, do you think it's the traditional way of getting things through regulatory bodies, FDA, et cetera? Like, are they more prone? Like, are there leaps and bounds steps that have to happen to make people think in different ways, to push funding forward, to push a trial forward, because you're doing it this way as opposed to the way it's been done for 40 years or whatnot?
I think that clinical trial design will continue to evolve. I think that there are two ways to answer that question. One of which is that certainly there are going to be new innovations. The way in which we have done clinical trials for pulmonary hypertension 10 years ago and 5 years ago may not be the way in which we're going to be proceeding in the next 5 years or the next 10 years. We hope that some of those aspects will actually accelerate the process, make things more efficient. I think that one example could be that, in the past, we had always thought that in order to get a drug ready for clinical trial, one has to test it in large animals in order to understand what the toxicity could be. Not only does that take time, it also takes a huge amount of money. However, in the new advent and possibly in the very near future, if we were able to understand and be confident that perhaps bioprinted tissue in a dish could actually serve as a surrogate in that regard, that could accelerate and may not make it needed to actually utilize large animals in this whole process as well. So that could actually be less expensive. It could also take less time. So I think that there are ways in which you can cut down the amount of time and the amount of money that it would cost to actually get any sort of new drug into practice. However, the other way to answer the question is that is clinical trial going to be obsolete in the future? And I would say no. I think that clinical trial is the way in which we know that a drug actually works, and we actually know that it has side effects that we can understand and appreciate, and they will not put our patients in danger. So I think that that's a very important aspect. I think that medicine has to be based on data in that regard, and I think clinical trials is the bedrock of how we actually do that. So I do think that we're going to continue to ensure that patients are only taking drugs that have been rigorously tested and has to be tested under conditions of a clinical trial. But those will evolve, and hopefully will become faster and more efficient.
Awesome. Well, people that are attending here, please take a moment if you have some questions either in the Q&A tab or in the chat function. And I'll start one that came in during your talk here, sir. It said, while prostacyclin analogs are developing continuously in terms of administration, and Winrevair has established a brand new leg in the treatment of PAH, in your estimation, what is the next paradigm in the treatment of PH or PAH?
So again, a couple of different ways to answer that question. I mean, I think that if this question is in regard to what pathways, what specific molecular targets are probably going to be the next paradigm, there are a lot of different opportunities and possibilities. I mean, certainly the idea that TGF-beta, activin signaling, and BMP signaling are part of what Winrevair is doing. I think that that is going to continue to be looked at, next generation drugs in that sort of scenario. We do know that BMP signaling and TGF-beta signaling are very broad and pleiotropic, and there could be ways in which there are pathways, offshoot pathways from those that are really important as well. There are other pathways, though, that I think that are important. I touched upon them briefly in terms of aging research. I do think metabolic pathways could be targets. We know that they're very important in terms of the development of pulmonary hypertension. I think that that could be a part of it. I very much believe in terms of anti-inflammation and chronic inflammation being a driver of the severity and mortality of pulmonary hypertension. So I think that new targets in that domain could very much be a part of it. I also think that there is an opportunity to look more carefully at the genetic and the genomic relationship of what our DNA does to predispose to pulmonary hypertension, and some of the pathways and the genes and the mechanisms that are entrenched in how our DNA encodes that. I think that those are very promising aspects as well in terms of the next targeting paradigm in this particular approach. I would also hearken back to the idea that there are ways in which we can understand the exposures that we have and how those can actually be implemented in ways that we don't typically think about. I love the idea of utilizing diet and exercise. And we know that the right type of diet, the right type of exercise training, we already know is really important and helpful in terms terms of improving pulmonary hypertension and using that as a treatment. How do we get that to a molecular level? How do we understand the molecular targets in a person's diet that are beneficial? How do we understand the molecular components of exercise that are important in this regard? I think that that's also going to be a really interesting possibility in the future of how we actually see treatment paradigms going. So that's a short answer, but certainly a very, very interesting process these days, and a lot of labs throughout the world are looking at this very carefully. I see
Yeah,
another.
here's a question. Yeah, here's a question from and I hope this is okay to say. This comes from a patient whose family was extremely involved in the earliest IV therapy clinical trials. And this patient asks, "Is there a safe way to have human tissue mid-disease to understand your black box theory more?"
Yeah, this has been a conundrum, a real riddle, which I think has been the reason why there has been a black box. Historically, the only way to get tissue is to actually biopsy it from lung tissue itself. And normally speaking, we don't think that that's safe at least from a clinical perspective, to do so in pulmonary hypertension. And that just simply has been, the paradigm and the dogma for which we've not had enough tissue in order to study. Now, there are scenarios here where there's a literature surrounding the idea that if we were able to get tissues from the catheter tip that we put into patients whenever we do a right heart catheterization to measure pressures and so forth, that hasn't fully taken off. And I think that there's a lot of controversy in that regard. So even that doesn't allow us a great satisfactory answer across the board in order for us to be able to understand that and to utilize that better. So I think that it has been a huge conundrum, and this is one of the reasons why I think that 3D bioprinted, I think tissue engineered solutions in this regard could be a really, really neat answer for us to be able to model disease even midstream. So the idea that, for instance, if we could print cells that we genetically manipulate that are coming from patients who actually are predisposed to pulmonary hypertension, and we print those cells in a way that it models more accurately what is happening in a person's lung tissue in and of itself. I think that that actually could allow us quite a bit of knowledge and information to understand what's going on at the early stages of disease, and really open up that area of discovery, and sure that this next wave of discoveries is going to be as fruitful as I think it would be. So I think that that's important. I think we have a follow-up question in terms of, wonderful, if at all possible, could you discuss epigenetic approaches? So the idea of epigenetics is a good one, but a complicated one. So epigenetics tends to refer to the idea that our DNA has certain intrinsic properties, the coding or the sequence that is always with us, never changing unless they're fully mutated, but is in every single cell. Epigenetics means that there's something that goes on top of that that actually can be changed, dynamically manipulated across different types of molecular, I guess, tags, as it were, towards changing the way in which the DNA is either structured or altered under that circumstance. That essentially leads to alterations of how cells express different proteins, and how those proteins then affect the way in which our cells become diseased. So there are a number of possibilities of trying to target those epigenetic changes on our DNA structure in order to improve pulmonary hypertension. There are ways, there are even drugs now that are being developed that can alter those, from histone deacetylation capabilities, microRNA or RNAi derived technologies. There are scenarios even for genetic editing, where we could use CRISPR-Cas9 editing or even base editing in order to change our genetic predisposition under these circumstances. All of those have not necessarily gotten to prime time time in PAH. I think that they are being investigated. I would say that, one of the caveats for epigenetics is that epigenetics tends to be a global process that affects a lot of different aspects of all different parts of our DNA. And so, if you were to manipulate that for a therapeutic benefit, it is not something that is easily as manipulated for a single spot on the genome. But like literally, if you were to change the way in which we alter our epigenetics in general, that puts you at risk, puts any person at risk for a lot of side effects downstream of how our cells just inherently process information. So I think that from a side effect standpoint, those have an inherent risk, but certainly could be very advantageous if we understand how to harness it appropriately. Um, Another one, could iPSCs that have been developed from PH patients be used for this type of research? Absolutely. So iPSCs are what are called induced pluripotent stem cells, which can not only be coming from and grown from patients with pulmonary hypertension with hereditary predisposition, but those can also be then genetically edited with technology that we have to actually edit back and forth. So like they are very fruitful in terms of discovery analyses. I love the idea of actually being able to utilize iPSCs from family members to be able to genetically manipulate them and even put them back into bioengineered tissues like 3D bioprinted apparatus in order to really understand at a molecular as well as a 3D reconstruction of the lung and the heart what is going on and how can we understand that biology better. So absolutely, I think it's going to be important. Our lab has certainly been very much a proponent of using iPSC technology in this space. They're not perfect, and so like they certainly may not reproduce every single thing that a person's cell in their own body actually does, but I think that it does do a lot in terms of helping to understand that genetic predisposition better. Additional question, could iPSCs from family members with genetic mutations who did not develop PAH be used to compare to those who have developed PAH to learn about why PAH develops? Absolutely. I think that, the idea of being able to use those in screening as screening tools where you could say that a person who actually has not developed PAH but has a genetic predisposition, take those cells and systematically try to alter them in the dish and see, what are the alterations that are required to change the way in which a person's cell goes from healthy to disease under that circumstance, and can be a great way for us to understand it at a molecular level. So absolutely, there are a lot of possibilities for that type of technology to develop over the next few years.
Um, how about what, all this great work you're doing, hypothesizing you're doing about your discoveries in the lab, what makes something like a promising discovery to a realistic drug target? And, how does that get to actually to a patient,
Yeah, it's a great question. I think that there are a lot of different ways that different discoveries can get to patients. And so part of it is based on the data and the strength of the data. So certainly we hope that the best data, the most robust data are the ones that actually get translated. It unfortunately doesn't happen always in that capacity. There's a little bit of luck that's associated with it, but we do hope that the most robust data is the one that surfaces. Now, when we talk about robust data, I do want to emphasize that there are different ways of actually thinking about what does robust data actually mean. In my view, I feel as if having what we call orthogonal pieces of data that all converge on the same point is really important. So that is to say that you may read a paper that shows in one model, like in a mouse model, this particular drug had a great effect. Okay. And that in and of itself is actually pretty exciting. What would make it more exciting, perhaps more robust, is the idea that there were data that accompanied that we now understand why and how that drug works. That drug works across multiple different platforms, not only the mouse, but also a rat, and maybe a 3D bioprinted reconstruction of a patient lung. Maybe there's a notion that there was computational modeling that went on that also was convergent in that regard. And what I particularly think is very valuable these days is to go and look at the human population of actual human patients with genetic abnormalities, and to say that in that scenario, do people with a genetic abnormality in the same target that is being affected by that drug, is that particular genetic abnormality also pointing us into that particular scenario? So that we have human data, we have mouse data, we have cell data, we have rat data, all of which pointing to that. I think that in and of itself is very important to look at, so that you have multiple different platforms independent of one another that all point to the same feature. That's much more convincing, at least in terms of scientific data. Now, even if you have all of that, it doesn't necessarily mean it even gets into clinical trial. The reason is that what has to happen from there is what we actually call the valley of death. And that's the valley of death of drug development. So a great discovery can often die in the lab because the people in the lab, people like myself and others, may not be drug developers. We've never been a part of pharma companies. We've never been a part of a clinical trial in that regard. So there are many scientists who are much more molecular who say, I can make this discovery, but I just can't make that leap over there. They need help. And that help is important because it actually means that a discovery is translated into a drug, and that drug is translated into a real program that gets into clinical trial, phase 1, 2, and 3 clinical trials and beyond. Oftentimes that takes a village. It takes more than just one person, a lot of expertise. It takes biotechnology, it takes pharma experience, it takes drug development experience. And it often, as I come back to as before, not often, it does take money, it takes investment, and so it takes lobbying too. So the fact is that patients with pulmonary hypertension, their families, their loved ones, it's really important to understand that process, that one discovery does not necessarily mean it's going to get into clinical practice. It means that we have to lobby as a community to say that we want these medicines, we want the development of these medicines to continue at a rapid pace in order to make sure that it gets into clinical practice and into the patients who deserve it the most. So it requires that. It requires you to tell your government representatives to say that we don't want funding for pulmonary hypertension to falter. We don't want that to go away. We want to make sure that we can get this moving and organized. So that part I think is really important. And because of that divide, because of that valley of death, it often is that sometimes the people who make the discoveries are not well connected enough to actually know, who do I talk to get this drug developed, who can I actually get help from. And so that may mean that we need to actually step in as a community to say, let's lobby and let's advocate for the most interesting and the most important discoveries, and let's get them moving forward.
Let me ask you about where we are at now with disease modification, or striving into that arena. How do we go from disease modification to disease reversal, and what does that look like for the community, or how do we
Yeah,
know if we've achieved that?
yeah. So, we've debated that a lot. I think that's come up with some of these next generation, new generation drugs. There's a lot of question as to whether or not Winrevair is actually disease reversing or disease modifying. It's hard to address that because, normally speaking, at a molecular level or a tissue level, we often think about disease reversing by saying that, well, if we knew that the blood vessels in your lungs were totally diseased and remodeled, if we gave you a drug and we were able to look at those vessels a year later or 2 years later, and we saw that there was regression, that there was actually a transformation of those vessels from being totally remodeled to going back to normal, that would be great proof to say that this is a disease modifying therapy. The problem is that we, again, don't have the ability to sample lung tissue from a person who's alive and well with pulmonary hypertension before and after medication. So that's not something that's going to be possible. So we have to use surrogates, and surrogates are things like, well, how do you feel? How far can you walk? What are the molecules in your blood doing? Those sorts of things. Does that help us determine disease modification? And probably it doesn't do so as much. Those are only very indirect measures. So one thing that we've actually been very interested in trying to figure out is that if there is a disease modifying therapy out there, is it possible that if you were to take that medication and get to a point where you think, wow, I feel great, I feel a lot better, I'm walking further, I don't have as many symptoms, do I think that this is disease modifying? Well, what happens if then you stop the drug and then you see whether or not over time, as you've been off the drug for a while, do your symptoms continue to stay resolved, or do they actually go back to what they were in terms of the height of your disease? So that could be one angle to look at. Now, I'm not advocating for anybody to go off their drugs, but what I am saying is that there may be data coming in the future to say that for people who had been on Winrevair for one reason or another and did well, could they actually, some of them have had to go off of Winrevair because of side effects. Well, maybe we could actually use them in terms of very important data to find out a year later, 2 years later, are their symptoms from pulmonary hypertension still resolved, still better, or have they actually gone back to what they were before? And so I think that some of those aspects are ones that we were going to look at very carefully in terms of clinical resolution, even off of the drug. Is that something that we are seeing with some of these medications? We hope that some of these medications will have disease reversing or disease modifying properties in the future, but so far we haven't gotten any proof for anything that right now, at least to that level, that there is truly a reversal in that space. But we're hoping that over the next few years, we're going to be able to figure that out.
Let me ask you, what kind of patients are the most likely to benefit from participating in early stage trials of emerging therapies? And I ask you that because this question has come up in a previous webinar with specifically with patients in Canada. There's concern, at least to this one patient, that are they too sick to get involved? Because does it mess with the trial that they're enrolling? So how do you balance that?
Yeah, I mean, every trial is different, and every trial should be strategized in terms of saying like how severe or sick is the disease for the patients we're recruiting, and a lot of them tend to be sort of, like I said, at least in the past, a little bit more one size fits all. I think in the future it is possible that we are going to be able to do a much better job of saying what are the markers that we can detect right off the bat that will tell us, does this person actually have a very good chance or not a good chance at responding to this very specific medication? That requires a new set of biomarkers that we don't have right now, but I think that could be in the future. I love the idea of genetic predisposition, to say that people who actually have genetic predisposition may have that marker, to say that these are the people that we want to target, to say this is the drug for you because it's targeting that exact genetic variant that you have, and therefore we think that you're going to be responding a lot better under those circumstances. So I think that that's going to be happening in the future. We haven't gotten to that point yet, but I do think that clinical trial design in the future can and should get to that point, because I think we're going to be able to do a much better job, particularly if you think that who's going to respond well. It means that the number of people that you have to recruit into your trial to see an effect is not going to be as many. So it means that those trials can actually go faster, can go quicker, will be enrolled sooner, and I'll get the data relatively soon, as compared to having to wait for years in that particular regard. I think that my own personal experience was that as Winrevair was getting approved, we knew that it was a really good drug based on what the phase 3 data was, but it just hadn't gotten approved yet. Had a couple of patients who were on the transplant list who were waiting transplant versus starting Winrevair. We had to do something because some of them were teetering on the point of essentially dying, and so we had to actually make a decision. And because it took long enough time for us to get Winrevair approved, some of those patients had to go to transplant even though they really, really didn't want to. They really wanted to find out whether Winrevair could actually hold that off. And that stuck with me. I mean, I think that I would hope that in the future we could actually be much quicker for the people who actually would be good candidates for a drug they could avoid something like transplantation. They could avoid death, of course. Those are things that we really would like to emphasize and make sure that we can actually get in play as soon as possible for the people who need it the most.
I know you covered this kind of broadly, but is there, if you had to point to one or two areas that you think aren't getting enough attention in or the investment right now, through patients, through industry, through scientists, advocacy groups, is that what are the one or two things that you would really think everyone should focus mostly on, Stephen?
Well, I mean, of course, I think that it goes without saying that some of these next generation drugs right now are the ones that would inherently get the most attention. I certainly believe in that. I certainly believe in those studies. My own biotech companies that we started with the technologies that we developed in our own study, we have a metabolic drug that's a glutaminase inhibitor that we'd like to start testing in PAH patients very soon in clinical trial, phase 2 clinical trial. Those are the ones that we would really like to get off the ground. Like I said, those actually take a lot of money, and some of them are not as richly endowed as some of the ones that already are embraced by the big pharma companies. But sometimes the ones that are not embraced by the big pharma companies are the ones that need help the most. We want to be able to test as many um, um, opportunities as possible to make sure that we get the most the largest portfolio of therapies offered to our patients. So that's one thing that I would like to emphasize. I think that the other thing that I would like to emphasize too is the idea that the biomarkers that we are searching for, the idea that we can actually begin to understand who is at risk for pulmonary hypertension, who may develop pulmonary hypertension, and can we actually identify this patient population, this disease as early as possible is a really important aspect because as we do that, the idea and the possibility that we can prevent pulmonary hypertension rather than just react to it is also going to be one of the new phases of the next generation, the next era. And can you imagine the idea to say that, before you even develop pulmonary hypertension, we could predict that you will have pulmonary hypertension in the future, but not only that, that we could give you a medication or give you a therapeutic approach to stave off that before you even develop any disease at all. And I think that that's something that is important. I think that the resilience to pulmonary hypertension is not something that we have talked about in the past. It's always been like, oh, you've got a fatal disease. We need to react to it, but it might be too late. How about thinking about it before it's too late? Let's make sure that we maintain your health for the decades to come. That's something that I think that we would love to be able to do, and I think that that's something that we should keep our eye on to ensure that it's a future that we can all see.
Well, that was basically my my last question, so I won't have you repeat that. I was, your future, looking at your clinic, so I deeply appreciate all your insights and the questions, the interactivity with everyone joining. A comment finally came in here. The future is exciting. I love proactive ideas, and as do we. So I thank you, Dr. Chan, and I really appreciate you taking time for us today.
Absolutely, and would love to be able to connect with all of you in person. Please don't hesitate to try to connect with me by email or hopefully in person someday soon.
And then if I could do just a moment of quick housekeeping as I click around here, to everyone joining, I really appreciate you joining us here today with this webinar series. I believe you see that there. We have one next week at 9 a.m. Pacific, at 12 p.m. Eastern. This is Global Centers of Excellence, Collaboration and Care with Dr. Benicio de Jesus Perez. And then on September 15th at 9 a.m. Pacific is Clinical Trials Matter, the backbone of innovation with Gene Elwing. I also want to say to folks, you can watch a playback of other webinars that we've had in the past couple of weeks here. They're available on our website at phaware.global/webinars. You can also find them inside the HeartWorks app. If you haven't gotten that, you can get that on your Google and Apple stores or at phaware.global/HeartWorks app. So I will put a playback of this. We'll have this up in later today or tomorrow for those who didn't join us. But otherwise, let me get out of here. Sorry. Stop sharing. So otherwise, thank you. And we will see you next time. And again, Dr. Chan, thank you for joining us today.
Thank you for having me.